The overall goal of the following experiment is to determine the impact of early life stress on the urogenital system by measuring perigenital mechanical sensitivity and mast cell activation within the prostate in male C57 black 6 mice that underwent neonatal maternal separation. This is achieved by first performing neonatal maternal separation on litters of C57 black 6 mouse pups to induce early life stress for 20 consecutive days. The second step is to measure perigenital mechanical sensitivity by application of a series of von Frey monofilaments to the perigenital region. Next, acidified toluidine blue staining is performed on cryostat sections of the mouse prostates in order to quantify mast cell activation. The results show that neonatal maternal separation increases perigenital mechanical sensitivity and prostatic mast cell activation based on our behavioral and histological analyses.
The main advantage of this technique over existing methods such as experimental autoimmune prostatitis is that it incorporates stress exposure during early development, which precipitates symptomology later in life, similar to what is observed in patients with chronic prostatitis.
This method can help answer key questions in the chronic pelvic pain field, such as the impact of early life stress on behavior and provide histological evidence of altered pain states.
Though this method can provide insight into chronic prostatitis, it can also be applied to other functional pain syndromes, such as interstitial cystitis, painful bladder syndrome, vulvodynia, fibromyalgia, or migraines, and can even be applied to other common psychological comorbidities such as anxiety and depression.